All right, I decided I needed a bit of a break. So now we're back to talk about the last bit of this week's information from Chapter 19, protein targeting and sorting. This is really section 19.5. All right, so there are three general groups of proteins. So proteins can either go to the endomembrane system, or they can be translated in the cytosol and remain there, or go to a different organelle. So we've got 1, 2, 3 general groups. So we're going to talk about this part first. All right, so proteins translated in the cytosol. This is where we talk about free ribosomes. Okay, so they're not on the ER, but they're doing translation. You got your mRNA. And if this is a category 3 protein that's going to go to some other organelle, it's going to need a signal sequence. Okay, so it has to have some information, and sometimes the signal sequence is in the middle or on one end or the other, but it has to have some kind of amino acid zip code so that it can get to its proper destination. So we're going to talk about proteins that are targeted to the mitochondria, the chloroplast. Okay. And in general, they have what we call a transit sequence. And that transit sequence is going to bring it to a receptor on the outer membrane of the chloroplast or the mitochondria. Remember that these organelles have double membrane, so they have an outer membrane and an inner membrane. And you've got Tom and Tim and Talk and Tick, right? And so these are where the transit sequence binds to the outer membrane of the mitochondria or the chloroplast or the inner membrane. So we're going to look at mitochondria because all eukaryotic cells have that. Chloroplasts only in the green ones. All right, so here you have your ribosome translating the protein in the cytoplasm, free ribosome. And we call this kind of location of the protein to the mitochondria post-translational import. Okay, so post-translational import means it's imported into the organelle after translation is completed. Co-translational import, you will see, is for the endomembrane system proteins. HSP 70, heat shock protein 70. This is also more generally called a chaperone. Right? So just like when you were younger, you had a chaperone taking care of you, making sure you were okay when you were out in the big, big world, right? Proteins have chaperones that make sure they fold correctly. Okay? And in this case, the chaperones are keeping the protein unfolded because you're going to have to wind that protein through the pore, okay, into the desired location. So you don't want it to be this globuled protein. That's not going to fit. Right? So the heat shock proteins bind to this protein as it's being translated. After translation is complete, the sequence signal binds to the receptor, and you can see down here the protein is threaded through that pore. Takes energy, not surprisingly, and there can be other heat shock proteins that are protecting this amino acid sequence until everything comes in and the protein can fold properly. So here's HSP60, okay, heat shock protein 60. So all these heat shock proteins are also what we call chaperones. Post-translational import means it's getting to its destination after it's been translated. So this would happen for all of these different organelles being targeted. Okay. Co-translational import means it's being put into the ER as it's being translated. So this is pretty cool too. So these proteins have this ER signal sequence on the N-terminus. Okay, so as the protein is being translated, you remember directionality? Proteins have an N-terminus and a C-terminus, and the N-terminus is what's translated first, the C-terminus is translated last. So there's this little signal sequence that this SRP, signal recognition particle, binds to. Okay, so this protein complex binds here, and it actually stops translation. And it stops translation so that it can take the ribosome and the protein and the mRNA to the ER, where it docks on this SRP receptor. And here is the pore where the protein will be translated through. So you can see here's the signal peptide. And now translation continues. And you can see as this purple protein is growing, it's being fed right into the ER lumen. So that's co-translational import. It's being imported while translation is happening. Here is a co-translational modification. Signal peptidase cleaves off the ER signal. Right? So that protein doesn't need that signal anymore because it's already been translated where it should be. Okay? We don't want it to go back to the ER or get stuck there. So the peptidase, which is a protein or peptide cleaving enzyme, just chops off that little signal. The protein folds, goes on to do whatever it's supposed to do. So this is an example of what we call a soluble protein. And we're contrasting a soluble protein with a transmembrane protein. Transmembrane proteins, like we've talked about before and you just drew in your last homework, have regions that span the membrane. So there are two different types, general categories. Type 1 transmembrane protein means that the N-terminus is in the ER lumen and the C-terminus is in the cytosol. Okay, so that's type 1. And so what's happening here, now they're not showing you all the details, but it's got the ER signal sequence, so it binds the SRP, and it binds to this translocon, and then as it's being translated, it comes across this red stop transfer sequence. And that tells the ER machinery, okay, don't feed through any more protein. And so it stops transporting, continues being synthesized, but that region of the protein will remain on the outside, and this region will remain on the inside. Signal peptide is cleaved off. This is actually moved out of the translocon and stays in the membrane. Type 2 transmembrane proteins just have their orientation switched. So in this case, the N-terminus is in the cytosol, and the C-terminus is in the ER lumen. Okay. And how this is happening is it has an internal start transfer sequence. So it's working here on the ER, and it comes across this sequence and says, okay, now let's start transferring. Okay. And so it feeds this whole protein through, and if it only has only one transmembrane region, then it stays just like that. We've talked about, and you've been drawing, proteins that have multiple transmembrane regions. So that works by having a combination of start and stop transfer sequences. And this is incorrect in your book. It says it's an internal start transfer. This really should be an internal stop transfer sequence. Okay, so this is giving you an example. So you could do start transfer, and you start transferring, and then you come to the sequence that says stop. So now translation is happening outside, and then you come to another sequence that says start, so you start feeding it in and then stop. And so you can imagine how you can make these multiple transmembrane proteins. Okay. You can have the N-terminus inside or outside, depending if it's type 1 or type 2. Okay. And the C-terminus inside or outside. So you can start with a stop transfer and then do a start transfer and a stop transfer and a start transfer. So you can get any kind of combination of these to wind that protein into the membrane. All right, post-translational import into the ER. Okay, so this one threw me because we've been talking about all this co-translational import. So I just want you to think of this as an FYI right now. Most proteins are co-translationally imported, but there are some that are actually translated in the cytoplasm and then moved to the ER. And the examples for this are primarily tail-anchored proteins. Right, so remember in your homework you were working with a lipid anchored protein. Okay. So if it just has its tail anchored, it may be translated in the cytosol and then imported into the ER. Another general class is some secretory proteins. These are the proteins that are secreted outside of the cell. What you have to know is every time we make a rule, and you can't see me, but I'm quoting rule with my fingers. In biology, there's always exceptions. Okay, so if you're interested in this, I do have a paper on it. I could be happy to send it to you, but this is really an exception to the rule. So we're going to focus on co-translational import when we talk about the ER. All right, protein folding and quality control. Okay, this is a really busy slide. This is not meant for you to know. I want you to understand that we have many mechanisms in our cell to get rid of a protein when it doesn't fold correctly. So all these little red circles are chaperones. And our cell has lots of chaperones that are trying to get proteins to fold correctly. And the reason is it takes a lot of energy to make a protein. It takes a lot of resources. So if the protein has the correct sequence and can be folded properly, the cell wants to put energy into that versus having to start over. But if a protein is folded incorrectly, there are many ways to get rid of it. Through the lysosome, through the proteasome, through proteases. And what it does is basically all these different methods chop up the protein back into the amino acids and recycle. Because you don't want aggregates. Okay? You don't want clumps of proteins in your cell. That can lead to disease. One of the general types of diseases that has protein aggregates as a problem are neurological diseases like Alzheimer's. Okay? You get these plaques in the brain, which means these cells have died because they get these protein clumps in them that don't allow the cells to function. So there's a lot of research on why do these proteins aggregate, why don't they fold correctly sometimes, and what could we do to fix that. So again, busy slide, just to understand that there are many mechanisms for making sure we make proper proteins in our cells. All right, and another busy slide to add, just to let you know you're learning so much information, right? You basically know all this stuff on this slide. We've talked about it, different parts. Protein trafficking. Okay, we're going to continue this in the second half of chapter 12, right? But General Biology 1, we learned we make proteins. Now you're taking that a step further and saying, okay, not only do we make proteins, but they have to go places, and they have to fold correctly, and they have to get modified and get to where they need to go. And that's really what this unit is about. It's about membranes, and it's about making proteins and getting them to the right spot. All right, thanks.